Welcome to today's webinar. We are going to give everyone a moment to log in and we'll begin shortly. Thank you. Thank you for joining the webinar. Are you sure about that ID? Ensuring microbial identification meets USP standards is presented by Abby Roth, founder of Pure Microbiology. Abby has over 21 years of experience supporting the testing and consulting needs of the pharmaceutical, medical device, and compounding industries. Her background in pharmaceutical microbiology includes extensive knowledge of environmental monitoring. Abby served as a USP Compounding Expert Committee member during the 2015-2020 cycle. She is an involved member of the Controlled Environment Testing Association, CETA, serving on the board of directors and speaking at its annual meetings. She is also an invited speaker for state boards of pharmacy and national organizations. Today's webinar is ACPE accredited for 1 contact hour. To receive CE credit, complete the CE evaluation found on the right side of the webinar platform. We are recording this session and will share the recording and evaluation link in a follow-up email. You can submit questions during the webinar using the Ask Question box on the right side of the platform. We will answer the questions submitted following the presentation. Thank you for joining today's session.
Right. Thank you so much, Amy, and to ARL Biopharma for having me today. I am really looking forward to talking about this topic, so welcome everyone, and we'll get started. So here are the disclosures. I have nothing to declare as far as conflicts of interest, so we can move on from there. This is the information then again about the continuing education credit. And here are our learning objectives for today. First thing, we want to make sure that you can come away from this session being able to explain the USP 797 requirements and also what is discussed in USP 1113 in regards to the definitions for microbial identification. We'll also hope that you can distinguish between phenotypic, proteotypic, and genotypic identification methods that will be used for microbial IDs. And then be able to kind of review a standardized checklist to assess and qualify microbiology labs for microbial identification. Just remember here, we are speaking specifically to how this relates to USP 797 sterile compounders when it comes to microbial identifications. I want to start by going to this quote that comes directly out of the chapter, and this is the requirement that surrounds everything that we need to be concerned with microbial identifications. So it does say if levels measured during viable air or surface, depending on which section we're reading this in, air sampling exceed the levels in Table 7, an attempt must be made to identify any microorganisms recovered to the genus level, and it mentions 1113, with the assistance of a microbiologist. Now that's a lot to unpack from this requirement in regards to microbial identification. So I want to go through each one of these pieces so that we get a better understanding of what's actually required and be able to apply some of that to what we do. So we're going to start off here by looking at the identifications in regards to how these apply to exceeded action levels with our viable air and surface sampling. So these are the action levels that we see in USP 797. It does end up giving us action levels for both air and for surface. For air, again, it's per cubic meter. For surface, it's per whatever device. So in this situation, you're seeing that it's reading per plate it really will kind of come down to what sampling device you use, whether it's a swab, a paddle, etc. I think we're all generally familiar with what constitutes an action level based on USP 797. The only thing I do want to call out here, though, is that FDA considers any microbial growth in the ISO class 5 area an insanitary condition. So if you are trying to work the insanitary conditions guidance document into what you do on a daily basis, you would strongly then want to consider defining that any microbial growth recovered in the ISO class 5 would be considered an exceeded action level. And this is really going to come down to your regulatory oversight and things that you need to consider as far as that goes. So when it comes to identifications, and what I would consider best practice is develop a plan that meets your needs. You could determine that your organization is going to do more than the minimum of the chapter, which is just to identify exceeded action levels. You could determine, hey, you know what, we're going to identify all microbial growth in the ISO class 5, even if it doesn't exceed the action level. Or you might have that in place for ISO class 5 and 7 spaces, but maybe not ISO 8. Or you could identify all growth as we had previously been required to do so. So, when it really does come down to what you want to do, you can base that on a couple of things. Categories that are compounded. Are you doing category 1 and category 2 only? Are you doing more complex compounding and you're going to be applying category 3 beyond use states? Are you doing non-sterile to sterile compounding? These are some of the things that you really could heavily consider in determining whether or not you are going to do more from an identification standpoint. I will also mention here the past trends note as well. If you've struggled in the past with a lot of microbial recovery, or specifically maybe for molds, you might benefit from having these organisms identified. All right, so the next requirement that's in this blurb that we see in the chapter is about an attempt to identify these microorganisms. And that's kind of a weird word for us to see in USP, but why does the chapter allow for just an attempt? And when we say attempt, this isn't like, oh, I packed up the samples to ship them out to the contract lab and I forgot to send them. That was my attempt. That's not what was intended by the chapter. And where this sort of comes from is when you ship your plates to the contract lab for identification, it might be when they subculture that media or subculture that organism onto fresh media, it might no longer be viable. So we've run into that in my past lab life where we'll get plates that'll come off test. There are microorganisms on there and we'll transfer them to fresh TSA plates. And when we do that transfer, they will not grow anymore. And without that, 18 to 24-hour culture, we were not able to perform an identification. If we do end up being able to get that subculture to grow out, there are some situations where the identification system that we'll use may not be able to identify the isolate. So where does that leave us? Well, it pretty much leaves us with getting some sort of information back from the lab that says something along the lines that, the identification was not possible. Here's the gram stain result, and here's maybe another descriptor. So for example, maybe they weren't able to get an identification to the genus level, and you're getting on your report something that says that it's a gram-positive cocci, and it is catalase positive and coagulase negative. Well, at least that's enough information for you to take some remedial actions and be able to maybe start a root cause investigation as to where that microorganism may have come from. So, this word attempt was really added to give you the flexibility that if the lab couldn't get an ID to the genus level, that you weren't somehow getting cited by a state inspector or an accreditor for not truly having a genus level ID. Now, the next thing, the microbial identifications must be done to at least the genus level. As one of my microbiologist friends would say, let's get nerdy again. And I want to kind of talk a little bit about what this means from a naming standpoint. So this is where we have the binomial nomenclature that comes into naming of organisms, whether it's plants, people, bacteria. We have two terms that are going to come into here, and this is really our formal naming system. The first name that we have is the genus, and the second name that's listed is the species. So if you are looking at your report and you're getting a result back from the lab that says Staphylococcus epidermidis, you're getting an identification to the species level. So in this case, genus name is Staphylococcus epidermidis is the species. Now you're like, why do I need to do this? Do I need a species level ID? That's not what's in the chapter. Well, it is going to provide you with more information when you have to do your investigation. So there are some major benefits to this for sure. It's going to help narrow the source of the microorganism. And a good example of this is that there are some gram-positive non-spore forming rod bacteria that when we get the genus name, we can't tell if it's from people or if it's coming from animals. But if we go to the species level, we can differentiate and distinguish that and better know that's a people bacteria versus maybe that's something that might be coming from livestock. So by getting a species level identification, it really could end up making your investigation overall a lot easier. And again, consider some of your risk factors here. Are you having a lot of exceeded action levels, especially at the same location with seemingly the same organism coming up over and over again? Species level ID might be helpful at that point. And maybe again, if you're getting a lot of recovery in your ISO 5, that really could warrant a species level ID. Next, it says microbial identifications must be done with the assistance of a microbiologist. And whenever I picture this in my head, I am seemingly seeing a pharmacist standing somewhere in the general pharmacy area and a microbiologist over their shoulder talking them through how to do an identification. So when it says assistance, I mean, you're really sending your samples out to a lab that can do these identifications for you. Whether it's going to be your clinical lab if you're in a health system, or if you're in an independent standalone pharmacy, there's a number of contract labs that could assist with this. When it comes to kind of choosing who you're going to work with, we'll get into that a little deeper a little later on, but I wanted to provide some information from USP 1117. And this chapter is the best microbiological best laboratory practices chapter. And it is informational in USP. But it does give us some very good information as to what sort of training and education should exist for those that are working in the microbiology lab. So in this case, the lab staff should have education, training, and experience to do their job. That really doesn't sound any different than what we're expecting of pharmacy staff. Their training curricula should be established for each laboratory staff member specific to their job function. I worked in a contract lab for a number of years, and not everybody does everything. So some people are trained on certain tests versus others. There's a hierarchy of different levels of lab technicians, microbiologists. So there should be a definitive set of requirements for an individual depending on what their responsibilities are going to be. And then the last one just indicates that there do need to be training records that are current and in place and show that these individuals were trained to the most current version of a particular SOP. So again, just ensuring that these individuals are up to date on training and have the appropriate background, education, and experience to do this work. Those are some of the things that you can specifically ask your contract lab if you're interested in kind of seeing what they have for a training program. The last one here to me is the biggest out of all of this, because it does say microbial identifications are going to be performed, and then it mentions USP 1113. So again, this is the microbial characterization, identification, and strain typing chapter. Yes, it's informational, but when we are directed to this chapter from 797, which is an enforceable chapter, that does hold a little bit more weight. So my question to all of you is, have you read USP 1113? And especially for those of you that are responsible as, the designated person for the viable sample collection and maybe investigations associated, this is really a critical chapter that if you haven't taken a look at, you really want to read. Now, interestingly enough, as I was working on this presentation, I went into my USP account and was like, oh, 1113 is out for public comment. And what you're seeing on the screen there when I took that screenshot is that it was 60 or 87 days that it was still going to be open. We're down to 68 days now that this is still going to be open for public comment. So for those of you, again, with your online USP subscriptions, even if you just have the compounding compendium, you can log in and access this chapter. And the cool part right now is you have access to the current version, but you can also take a look at the proposed changes that the Microbiology Expert Committee is looking to implement. So it's a really interesting read to see kind of where we were and some of the changes now that we are expecting to see. So I just want you to take a second. When you hear the word identification, what comes to mind? What do you think of? And I asked this in a class that I teached recently, and I got a couple of things that they gave as answers. It was kind of along the lines of, well, it's a name, it's some sort of like microorganism, it's a test that they're going to run. And there wasn't really like a clear definition that these compounders could give to me of what they really thought of, when they hear the word identification. So I want to share with you the definitions that we get from USP 1113. And what I did here was took the current definitions for both identification and characterization and the proposed definitions. So we're going to go through all of these. I know this is a heavy worded slide, but it's really key here that those of you in the pharmacy space really understand these differences. So when we talk about the term identification, and the current definition that we see in 1113 says the determination of which a broad group, so is it a bacteria, yeast, or a mold, or a narrow group, genus and or species, to which a laboratory isolate belongs. So that definition is like, okay, when it comes to what we're really looking to do for USP 797 compliance, just saying that it's a bacteria, yeast, or mold really isn't meeting our needs for identification. We really need that genus or species ID. So it's really nice to see that the proposed version of 1113 kind of takes out that broad group and really focuses now in on an identification is a genus and species level ID using phenotypic, proteomic, or genomic methods. So I'll go through those categories a little bit later, but we are definitely using some more advanced technology to actually get a genus and species identification. Now, this is where things get a little murky, where we start talking about the term characterization. And looking at the current definition of 1113, characterization is saying, okay, this is where we're looking at colony growth. So what does it look like on the plate? Cellular morphology. So when we do a stain of it or look at it under the microscope, what are we seeing? And then other key diagnostic features. So there's some other tests that we could run that could generally categorize a microorganism without actually doing a full identification. The proposed definition is very similar. You'll see the same kind of language there in the first half of the definition. The nice part here is it added a little bit as far as examples. And doing gram stain results, cell shape, the arrangement. So again, if we would see something that was a gram-positive cocci in tetrads, yeah, those of us that are on here that have microbiology backgrounds, we're like, well, that's micrococcus probably. More than likely, we could guess that that's what the genus name is going to be, but we didn't actually do an identification. So I want you to understand that there is a big difference between microbial identification and microbial characterization. And we're going to continue kind of this discussion as we go here. So last note before I leave this slide is that what we need for USP 797 compliance is this identification to the genus level at least, and many of you might benefit from that species level ID. The other awesome part about 1113 and what they're looking to add, and fingers crossed that this goes through, because remember, this is proposed, so this might not be what the final version looks like, but there's this great table that breaks down a whole different slew of like processes that we're going to have microbial samples for and maybe need to consider identification. So you'll see here for CSP release testing, and the type of test we're talking about is the USP 71 sterility test. This chapter is stating that if there are any microorganisms that are recovered, they need to be identified. So that's kind of where it's the, well, microorganisms to identify any that are recovered, and what we should do for the identification as the minimum recommended is an actual identification genus species level. So if we look at the next group down then in the white with the viable sampling, any growth that is recovered in the ISO 5, this particular chapter will be recommending that it is identified. If there's any exceeded action levels for ISO 7 or 8 areas, it also recommends identification. And then not in our world, but in the manufacturing world, we'll say some of them have alert levels. So in this case, it's recommending that if there was an exceeded alert level in an ISO 7 or 8, that it would be characterized. So the really great part about this is that what is proposed to be in this table is actually going to match what we see in USP 797, at least for the sterility testing aspect and for exceeded action levels in ISO 7 or 8. For ISO 5, again, this would leave it up to you to do more than what 797 requires, but there is, I think, benefit there, especially if you're seeing continued microbial recovery in the ISO 5. Doing an identification would definitely be beneficial. So I'll be very interested to see where the microbiology committee ends up going with this table based on the public comments. But I really do think we're probably going to end up with a really helpful informational chapter. So that now leaves us with, what are you getting from your lab? We just went through what these definitions are, so maybe some of you are thinking, well, where does that leave me? Because I'm not really sure what I'm getting from my contract lab. All right, so we're going to get into now some more of the sciencey parts of this, not too deep. We'll try to keep this all understandable for everybody, but the identification method that the lab uses actually does matter. And we have the 3 categories that are mentioned in USP 1113. So the phenotypic, the proteotypic, and then looking at genotypic. So when we look at these at the bottom of the pyramid, that is kind of giving us that broad identification where the accuracy might not be as great, nor is the reproducibility. But as we kind of funnel to the top of that pyramid, the accuracy gets better, and so does the reproducibility. All right, so when we're looking at these methods and we're like, okay, so some of these are going to be better than others, some of them are going to give us better IDs than others, how does that affect us? So looking at the right-hand side here, and again, this information comes from 1113, the greatest impact to us if we are given the wrong genus identification. And from that standpoint, again, that can be a pretty broad group of microorganisms. And if it's wrong, we start now going down the investigational path and are going in the completely wrong direction. We're looking for the wrong types of sources. So when we potentially get a wrong species ID, that's a little bit less of an impact on us. Again, we have these exceeded action levels. We need to do an investigation. We don't want to waste our time. But then the least impact to us is actually getting no identification. And that sort of seems counterintuitive. But ultimately, if we're left with a characterization at that point of, hey, it's a gram-positive cocci, we at least can kind of guess where that might be coming from and start our investigation. It's going to be broad and it's not going to be very specific to start, but hopefully we can funnel that down a little bit. So, these are some good concepts and things to think about as we're looking at these methods. So now's kind of the difference of what we're actually seeing here. So when we're talking about phenotypic, this is going to use expressed gene products and maybe some other metabolites that we're looking for. We need a large number of cells in a pure culture in order to be able to run this. And this would be an identification method where that lab is going to have to transfer your colonies onto a fresh medium to be able to get organism to work with. Getting an accurate ID here does require the selection of what it's saying a correct panel. A lot of times there's kind of a set panel of biochemical tests that we will run for these particular microorganisms, and based on the outcome of all of these tests, it will give us an identification. But if you don't properly gram stain the organism and then run it on the wrong panel, you're not going to get the right identification. So there's a lot of nuance that has to go into this to ensure that we are going to get an accurate ID. One of the other challenges here is that some of the stress microorganisms may not be correctly identified. So, if that's the case, we could face some challenges there as well. And depending on the panel or the identification that has to get run, there might be some other supplemental biochemical tests that the lab will have to do in addition to the actual identification. And the ones that are listed here, those are usually kind of pretty quickly accomplished. But from an ID turnaround standpoint, some of these IDs could take a couple of days to get an identification back to you. Now, the proteomic or the proteotypic, as you might have heard it called, this is going to be an analysis of proteins. In many cases here, this is going to be a MALDI-TOF system. So if you've heard that term before, it's a mass spec system that is going to analyze some of the proteins that we are seeing in the microorganisms. The nice part about this is it is a quick turnaround time on the identification, but it's still going to be a culture-based method. So again, that subculture onto fresh agar is going to be necessary in order to be able to run this identification. If you've ever heard of strain typing, this is not going to meet the needs for that, but that's pretty much more than what we in pharmacy are going to need for identifications. And the last note that you'll see here is that accuracy is all dependent on the database. Now, with these MALDI-TOF systems, the different manufacturers have different databases for microorganisms, and the lab has to be using the proper database. Some of them might be more of a research database. Some might be, again, more for like the clinical microorganisms that might be recovered. So that's a key piece to this, that the database is also accurate and appropriate for what we're going to be running. The last part of this are the genotypic identifications. This is the DNA sequencing. So we're going to get a really highly accurate identification. It is going to be a quick ID. Again, accuracy will be dependent on the quality of the sequence, the database, and then next steps beyond this would be like strain typing. That's way beyond what we need in pharmacy. Even the DNA sequencing is beyond what we need. But I want you to know that these things are available and out there, that, if you do feel stuck where you've been trying to get a microbial ID for something and it's not working, can't get it, you do have some kind of steps up the chain to do a more advanced identification and maybe hopefully get an ID. So I wanted to just share with you kind of some of the behind the scenes things that happen in the lab, just so that you get a feel for what goes on, because this is not usually simply like, oh, just put it on the piece of equipment and run it. With the identifications for bacteria and yeast, again, there's usually some sort of subculture to fresh media, unless we're doing DNA sequencing here. And that might be TSA, it might be TSA with blood. It's really going to depend on the identification system and the organism that we're running. That subculture is going to go in the incubator for 18 to 24 hours, depending on the system. And then there's some other things that we might have to do in order to be able to do the ID. So gram stains, auxiliary tests, if we're using the MALDI-TOF, gram stains aren't important. If it's a phenotypic identification, gram stains are key here. Then we can prepare the ID, run it on the system. And as I mentioned, there might be some additional auxiliary tests that need to now happen post-ID. And again, this is usually for the phenotypic methods. Now, mold's a little bit different. If you've ever talked to your contract lab about the mold identifications, and they're looking at under the microscope and they gave you a result of sterile white hyphae or non-sporulating mold. Well, that means that they're putting it under the microscope and looking for reproductive structures. So one option that we do in the lab is use the microscope to do the ID. So we can look at it macroscopically, and we're looking for things like, what does the surface look like? What color is it? What does the backside of the colony look like? Colors are important here. And then we can microscopically observe it as well. So we can create a slide looking at the hyphae. We can look at reproductive structures. Are we seeing branching conidiophores? What do the conidia look like? Are they round? Are they in chains? So there's a lot of pieces to that the microbiologists are going to look at in order to get a mold ID. And if we don't have those reproductive structures present, guess what? That's where you get the non-sporulating mold or sterile hyphae or something along those lines. Now, there's also an option for identifications on a system as well. That is a little bit more limited than it tends to be with bacteria and maybe with yeast. So that's a good opportunity for you to talk with your lab and understand what the identification options actually are. So what you might see on your report, if you are seeing things on your report like what are listed here, you're getting genus and species level identifications. It's an easy giveaway because both names are listed. So seeing, again, common organisms like we would typically come across, Micrococcus epidermidis, Micrococcus luteus, those are pretty common that we will see. Even some of the Aspergillus mold colonies, and there's a variety of different species that you might see, versicolor, fumigatus, brasiliensis, like, again, a number of different organisms. So if you're already seeing something like this on your report, you're probably in good shape. Now, when we're talking about characterization and screening, as I kind of had alluded to, they might do a gram stain. So what we're seeing in the picture on the left are the two different stain categories. This is all based on the cell wall structure of the bacteria. And if it's gram positive, it stains purple. If it's gram negative, it stains pink. So that's kind of the way that we do that differentiation. And then we also look at the cellular morphology. Are they circles? So are they cocci? Are they rods? And it gets reported along those lines. Then there's some other biochemical tests. Example here is showing coagulase, where that's used to determine the difference between some of the different Staphylococcus species. So with some of the other biochemical screening that you could come across, they could be doing an oxidase test. We use that to differentiate gram-negative bacteria. If it's catalase, it's to differentiate Staphylococci from Streptococci. And then coagulase, as I said, will kind of differentiate between what we would consider more pathogenic Staphylococcus and less pathogenic Staphylococcus. So, those are some of the things that you might be seeing on your reports. Or if you're just simply on your report seeing something listed as Staphylococcus, Micrococcus, Cariniform bacillus, gram-negative rod, yeast and mold. Those last three, obviously, we don't have a genus level ID. But when you look at these types of results, and if this is what you're more seeing on your report, these characterizations look very similar to genus names. And many of the contract labs might be putting Staphylococcus on your report because under the microscope, morphologically on the agar, based on a couple tests, we can generally put it and say that's probably the genus that it belongs to. And again, for the microbiologists on here, we can generally pretty confidently say that, but we didn't actually run a microbial identification as it's defined in USP 1113. So, you got to kind of be cautious about what you are seeing here. So what I wanted to share is kind of the identification versus characterization, and the middle column there is the morphology of the organism. And you're going to see here that if it's a Staphylococcus epidermidis, we would characterize that as coagulase negative Staphylococcus. And then these are all the morphology traits that would kind of come into play here. And there's not really a need for me to, go through all of this here. This will be a great slide for you to reference later on, but, there could be a number of different genus and species that meet these morphology bullets and then could be categorized generically as what you see in the right-hand side. So again, just trying to bring awareness here of what you actually might be getting from the lab. So one way that I was thinking about this as I was putting this presentation together, I'm like, categorization and the characterization is sort of like the city. Staphylococcus. That's pretty well, that's what we got going on here. We know that's what it is. And I see the identification as like your house number or your address, where we're going to get you to that final location to exactly where you need to be. So if you can kind of think of it this way, the characterization gets us close. The identification confirms what it is and gets us to that actual sort of final destination if we were kind of talking about directions. So I wanted to just take a second here now to run through a regulatory review. So if we go back to USP 797, it tells us, okay, you have an exceeded action level. You need to identify to the genus level. And then it tells you to follow USP 1113. When you go to USP 1113, an identification is defined as some sort of phenotypic, proteomic, or genotypic identification. So this is where, from a lab perspective, they have to be running an ID in order to meet the chapter requirements. If they're simply doing a stain or two and maybe another chemical test, that is characterization, and that does not meet the identification requirements that we are looking for. So now trying to find a lab that is going to be able to meet our needs for doing this particular work. First thing you want to make sure of is that there is a microbiologist on staff, and maybe that sounds silly, but there are labs popping up all over the place. And what sort of credentials do these individuals have to be able to do your testing? So ask for education, ask for training, ask for any credentials. I know there are a couple, lab people on this webinar today, and they will definitely back me up on this statement that they are going to be more than happy to provide you with any of that information. So anything that you need as far as that training, education, credentials, they're happy to supply that. Another piece to this that will kind of help in your journey here is the lab ISO 17025 accredited? So this is the ISO standard for laboratory and testing, so calibration and labs. This helps ensure a robust quality system. So out of anything, it makes sure that there's separation of quality and operations There's an independent group that's going to review results and give you some assurance that the work that is being done appropriately, ethically, and to meet the standards that are there. So with that, I would recommend that you ask to see the laboratory's scope of accreditation. Again, they are going to be more than happy to share this with you, and many of them already have that available on their websites. This is an example of a scope from a contract lab, and many times then too, this information is available on the website of the accrediting body, so you could just go look it up as well. So I'm going to zoom in here on the next slide, but I just kind of want to give you the layout of what this sort of looks like. It'll be a full page certificate, have the lab's information on it, and then it'll have all of their accreditation information. So in this particular one, what we're looking at here is biological testing, and then it's further defined out as viable air and viable surface testing. The accreditor that came in to check them out was specifically looking at their ability to count colony forming units and to also do identifications. The standards that they were holding the organization to were USP 797, USP 1116, and then also an ISO document, which is on environmental monitoring, which is 14698. So what you're seeing in that last column is the process that they use to do that testing. So enumeration is done by a visual count, meaning that there's somebody physically standing there counting all of the plates. And then their identification is done using a multi-TOF. So if your lab that you are working with is accredited and has a scope of accreditation, that's something that you can look for on there as far as the method or the technology or technique that they're using for identification. And again, ask for this. They are more than happy to provide this information to you. So the next one is, does the lab have experience with and knowledge of USP 797? And the first bullet, when I was drafting this presentation, I'm like, that sounds terrible, that there are labs looking to take advantage of compounders. But I do find that we are an industry that has a lot of testing needs. And there are some labs that try to break into this group that don't really have the knowledge base of the chapter that's needed. And it's very interesting because we do work that is most closely suited to aseptic manufacturing. But what we don't really follow here is the GMPs. So if there's labs that are going to, service pharmaceutical manufacturers doing some of the same tests that we need, they really might not know what's in USP 797 or understand what the compliance needs are. So as you are maybe looking for a lab partner or looking to change lab partners, you really want to talk with kind of their technical services people, their quality individuals, those that are going to really have the knowledge base of the testing standards that you need to have met. And then the last one here is an appropriate identification method used that can provide a genus level ID. Ask about their methods. So if you're starting to get into a conversation and you are not getting information that they're running it on a system like a multi-TOF or a Vitec, I'll even go back to like the MIDI systems for fatty acid analysis. That should be an immediate red flag to you that you might not really be getting a true identification to the genus level. They're starting to tell you that we're going to look at a wet mount slide or do a gram stain and then do a biochemical test. That is a characterization and not a microbial identification. So I'm really trying to drive that point home of spend the time talking to the lab, ask the questions, get somebody on the phone that will answer you. And it's not going to be a salesperson. If they give you a salesperson, please ask for somebody else to talk to. You want to talk to somebody that can technically answer some questions for you. and my lab friends that are on here, don't get mad at me for that. But we want to make sure that the compounders are getting what they need. All right, so to kind of summarize everything that we've run through today, USP 797 does require that exceeded action levels be identified. You can always do more if you want. And when we look at the kind of trail of what an identification actually means through 797 and into 1113, characterization does not meet the requirements. We truly need that identification to at least the genus level. If you haven't read USP 1113 and you are the designated person responsible for all of this aspect of the compounding responsibilities, take a look. And actually right now, I would encourage you to not only look at the current version, but also check out the proposed version as well and kind of see where we are going. Talk to your lab. If they are accredited, ask to see their scope. If you're not seeing the testing that you'd expect to see on their scope, again, sit down with them, ask a little bit more about maybe why they didn't do accreditation for that particular type of work. Are they planning on getting accredited? And if they're willing to send you a copy of an SOP, that's great. You can take a look. Sometimes they're not, which means it's time to go do an audit. If you really feel that this is something that you want to take a deeper dive into to kind of assure that you are getting exactly what you need. Okay, so thanks for hanging with me through that. I do see that there are a number of questions available, so I am going to hop into that question bank and start answering some of those. So the first question was, it says, is that statement in the insanitary guidance? So I'm assuming that that is the any growth in the ISO 5 is considered an insanitary condition. The answer to that is yes. You will find that in the FDA's insanitary conditions guidance document. And just remember that that document applies to both 503A compounding facilities and 503B compounding facilities. All right, next. Would trending be affected if different identification methods are performed, such as characterization versus genus identification? This is a fabulous question. And the answer to this is maybe, and probably more maybe on the yes end of things. If you've been doing characterization, again, if you've been working with a good lab, they should be able to get you pretty accurate as far as the grouping. But there could be some issues or errors with what was there. And we might start seeing some different microorganisms pop up when you are finally getting the work done to the genus level. However, remember kind of what I talked about, that no identification is actually okay. So if we're kind of thinking about that and you're going back and trending your data as a whole, if we're seeing, again, that these organisms that were characterized are coming from similar sources as now what was identified, it's likely not going to affect you too much. So I don't like giving a maybe answer, but it's probably really going to come down to what organisms that we happen to see based on that individual facility. That was a great question. So this one I'm not quite sure, but I'll read through it and maybe try to answer. It says, is it the only standard method for identification? So again, there are a couple different methods of identification. The use of the MALDI-TOF is not the only one out there. Again, there are DNA sequencing options. There are some phenotypic options that are available. bioMérieux has a couple of those that some of you might be familiar with. So there's more than one way to get an identification to the genus and the species level, but just remember that we are going to have a variation in accuracy and reliability with those different methods. All right, next one. Oop, things are jumping here. Hold on. All right, so it says, what do we do with the identification? What IDs are red flags? Well, so you are required to get an identification when you exceed the action level, which means that if you've exceeded an action level, you're lost potentially your microbial state of control within that environment, and you're required to do an investigation. So you're going to take a look at those microorganisms and where they've come from, what their potential pathogenicity risks are for individuals, and kind of piece together your initial corrective actions that maybe you're going to take because of the recovery, and then also look at your long-term corrective actions that need to be taken to eliminate the possibility of these microorganisms being recovered again. So things that I might consider identification red flags would be a lot of gram-negative recovery. I know we have sinks in our clean rooms. That's a potential source of it. But if we're starting to see gram-negatives make their way into the buffer rooms or into the ISO class 5, that's a red flag. Mold that is, again, migrating into the buffer rooms or the PECs. So seeing mold in an anteroom, I'm not terribly concerned because the anteroom is a buffer to the buffer room. And as long as we're not seeing that migration, it's not too much of a concern. So, you know, even if we're seeing significant recovery of spore-forming bacteria, that will commonly come in on a variety of different sources, but commonly it would be cardboard, soils, plant matter. So again, looking at people's shoes, clothing, how's material transfer. So those are some of the red flags that I would look for. Okay. So the next question is, again, these were, why is this jumping all over? How likely is misidentification with DNA analysis between species in the same genre? It is more unlikely of that. It's not typically a common issue that does occur. So I would say not very frequently. More likely going to see maybe more challenges, definitely more challenge with the phenotypic identifications, but even some of the MALDI-TOF could have some problems differentiating between species. The nice part about using systems like the MALDI-TOF, though, is the printout that you're going to get is going to give you some confidence levels as to whether or not it's a good identification. And your contract lab could answer some questions in regards to that. This one says, can you recommend additional coursework for compounders wanting to understand which organisms are considered more pathogenic or objectionable in the ISO 5 sterile compounding setting? So I can't think of anything specifically off the top of my head that's a good place to go for that. What I will tell you, though, is some of the contract labs are actually providing monographs of microorganisms when they do your identifications. So the nice part is that some of the labs, the microbiologists are doing the hard work for you. And, you know, at that point, you'll have sources, some pathogenicity concerns, and, you know, can sort of work from there. So if your lab's not already providing that, it might be something that, you know, you could ask them for maybe on a case-by-case basis. All right. Let's see. It says for ISO 8 viable air sampling, is the action level of one, so this is less than 100 CFUs. It should be greater than 100 CFU applied to each individual sample plate, or is it based on the total of all samples collected in the ISO 8 area? So that is per individual sample plate. All of the action levels that you see in USP 797 are per plate and not per all of the plates in a particular area. All right, let's see. It says, how often have you seen a microorganism identified as an ATC strain as would be used in the lab for method suitability or for sterility? So I worked in a contract lab for a very long time. That is not something that had happened in our world. Now, we did have kind of segregated areas of where we handled cultures versus where our identifications actually got done. So there really was a clear separation to try to avoid any risks as far as that goes. Hopefully that answered the question. Next one, have you noticed a reduction in organisms since the launch of the 2023 chapter? Overall microbial recovery, I have not seen an overall change with that. Although I will kind of say that for locations that really have maybe tried to ramp up their viable sampling program and do more of a risk assessment for choosing locations, I have seen initially with those groups there be some more failures, especially on some of the samples that are either in the PEC or in close proximity to the PEC. And I think for those groups that have really been trying to do more of a risk-based approach and choose some better locations, they initially saw some higher recovery, but it gave them the ability to take some appropriate corrective actions and make some changes to prevent those situations from reoccurring. So this said, just to confirm, biochemical tests like oxidase, catalase, and coagulase cannot be used to determine genus level. Even if it's done in-house, it would not be appropriate. So yeah, again, the way that things are reading, we are looking for a full phenotypic, proteotypic, genotypic identification, and simply doing those biochemical tests will not meet the chapter expectations of a full identification. All right, I think I have one more question and we're almost at time. So I like how this starts. A little tangent into media fills. Okay, I'm happy to answer that. USP 797 states to incubate final containers at 20 to 25 and 30 to 35 for a minimum of 7 days each. From a microbiological standpoint, is there a preference of which temperature to start with? Are you likely to have more growth when starting with one temperature versus the other based on growth rates of mold versus bacteria? Okay, so again, my microbiology friends on here will probably agree with this. We can go a number of different directions with that. So it really will kind of depend on kind of your stance on things. FDA's guidance document, which is the for aseptic manufacturing, tells us to start at 20 to 25. That will help with some of the environmental isolates that we might have some concerns with, especially with the molds in this particular situation. I also recommend following FDA's guidance document for that. So, all right, I think I got to everything and we flipped to the claiming CE credit. So I'll turn it over to Amy.
Wonderful. Thank you so much, Abby. This was a fantastic session. And thank you to our attendees for attending online today. We appreciate the Q&A. It was great. As a reminder, to claim your CE credit, please complete the evaluation link. This evaluation link is found in your webinar platform on the right-hand side. And then you can also screenshot this QR code and submit that evaluation as well. If you have any questions or concerns or require further assistance with regards to your CE credit, please email cesupport@acainfo.org. Thank you so much, everyone, for attending, and have a wonderful afternoon.